All right, so we are going to move on to the topic of genetics and Mendelian genetics because there are some laws that genetics follows, and then we'll also figure out how to do some crosses and how to calculate the percentage chances of what offspring are going to look like or what genetic combinations that they'll have. So the learning objective is that y'all be able to define genetic terminology and perform monohybrid crosses by completing notes and practice problems. So we're going to go through these questions as your notes from this video, and then we will do practice problems in the interactive notebook. So the first question I want to think about as an introduction to this unit would be, there's construction going on, would be, why do you look like your parents? And the hint is to think about, what did you receive from them? Okay, so you're going to answer that question, and I'm kind of curious what you guys think because, as you know, the title of the unit is genetics, so you want to think about something along the lines of with DNA. Okay, so we're going to watch this little video to preview the genetics part. Let me share the sound.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
All right, so that's a general overview of genetics and what we're going to be learning about and doing during this unit. So why do we care about heredity? Well, heredity is the passing of characteristics or traits from parents to offspring. It explains how and why you got your characteristics. So basically, like they said, every single trait that you got from your parents, you got half of it from your mom and half from your dad. And sometimes one of them is dominant over the other, right? One allele from either your mom or your dad is dominant. So that's the one that shows, and that's the one that you have, which is why you look kind of like a mix between your parents, because usually your parents have a mix of dominant and recessive alleles for a trait. Gregor Mendel was the man who discovered the laws of genetics that most organisms follow. So he was a monk born in Czechoslovakia in 1882, and he's known as the father of modern genetics. He was interested in understanding how genes are passed from one generation to the next, so he observed the pea plant. And the reason he did peas is because they're easy to grow, they grow really fast, and they can be self-breeding, and you can grow a lot of them at once. So they also have characteristics that are easy to define. So there's like 2 characteristics for every trait that he had in his peas. And so he always was able to figure out which one was dominant and which one was showing really, really easily. So for example, there are yellow or green seeds, like they showed in the video. There are purple or white flowers. There are wrinkled or smooth peas. And then there are tall or short plants. So all of these things are examples of traits that have this or that. And they're all dictated by one single genotype or one single allele, or a pair of alleles, sorry, on chromosomes that make it really easy for us to define what is showing up in the genotype of an organism. So true breeding peas. By controlling fertilization between plants, Mendel was able to create some true breeding pea plants. So a plant that's always true breeding will always produce the same offspring that look like itself. So if it's true breeding for purple flowers, it will always produce babies that have purple flowers. So then he took 2 true breeding plants with different traits and he breeded them together. And so he put true breeding purple flowers with true breeding white flowers. And he wanted to see what's going to happen with these. And when he did this, his surprising result was that all of the offspring between the purple true breeding plant and the white true breeding plant is that all the babies were purple. So what he was able to get out of this was that purple was the dominant color or the dominant allele that covers the white allele. So here's some key vocabulary. We know what a trait is, kind of like almost like a character trait, right? A physical trait in this sense is a characteristic or something that you can define the looks of an organism with. So for example, in humans, you can talk about having brown hair as a trait or having freckles or being short. Heredity is the passing of genes from parents to offspring. So when parents give their genes or half of their genes to their next of kin or their babies, that's heredity, right? You're receiving genes from your parents. A gene, on the other hand, is the basic unit of heredity. So it's the singular individual units of what your parents are giving to you. So they're located on specific points of your chromosomes, which are made out of your DNA. Alleles are various forms of the same gene. So like we said, for the flowers that Mendel was working with, there's one gene that has 2 kinds of alleles. And those 2 alleles are 1, purple, and 2, white. So they're 2 versions of the same gene, right? They're both coding for flower color. They both have the same job, same gene, same job, flower color, but they're 2 versions. So one of them is purple and one of them is white. Dominant versus recessive is important for us to know too, because if something is dominant, it's the stronger allele, and it's usually represented by a capital letter. It's the one that's going to show if it's paired with a recessive allele as the physical trait instead of the recessive one showing. And the recessive, obviously, on the other hand, is the weaker allele, so it's represented by a lowercase letter of the same kind of letter, because remember, one gene is represented by one kind of letter, and then that letter will have a capital or a lowercase representing if it's dominant or recessive based on which one is showing. So you'll only see the recessive trait, like I said, if there are 2 recessive alleles. So if it's lowercase, lowercase, or homozygous recessive, then you'll see that physical trait of the recessive allele showing. So I'm going to ask that you pause the video and go ahead and do the practice problems to define if the capital K is dominant or recessive, if the lowercase r is dominant or recessive, if the lowercase p is dominant or recessive, or if the capital F is dominant or recessive. For the phenotype versus the genotype. Phenotype, if you look at here, is the P is in red. Phenotype is the physical trait that you can actually see and is determined by the genotype and the environment. So for example, if somebody's hair is brown, that is a physical trait, and that's a phenotype. We also know that, for example, technically you could dye your hair, so it's not based on your genotype. And if you dyed your hair bright blue, no genes are ever going to code for that. So that's something to take into consideration when you're talking about phenotypes. On the other hand, genotype, begins with a G, is the G combination. So G, gene, G, genotype. It's the gene combination that determines the phenotype or determines what the physical trait is going to be. Remember, we talked about the central dogma. So the genes are the DNA. You're going to use these genotypes to make your mRNA, and your mRNA is going to translate into your protein, which is the phenotype or the physical trait, right? PPP, protein, physical phenotype. Okay, so we have the genotype where you're talking about the letters with the alleles of the capital R, lowercase r. In case of hair color, it was brown, capital B, lowercase b. So I want you to identify for the following example. You're going to pause after and tell me, is the capital A a genotype or a phenotype? Brown eyes, is that a genotype or a phenotype? Capital T, lowercase t, is that a genotype or a phenotype? And red hair, is that a genotype or a phenotype? Now we're going to talk about new terms called homozygous and heterozygous. So homozygous, like in the video said, it means that the alleles are the same. So whenever you have your two alleles, it means that they are the same type, whether they're both capital or they're both lowercase. So in this case, you can have homozygous dominant, which is capital R, and you can also have homozygous recessive, which is lowercase r. Okay, they're homozygous because the base word for homozygous is homo, which means of the same kind. Right? So there's also other terms, for example, like homosexual, which means liking the same gender or the same sex, right? Homozygous means that they are of two of the same alleles. So again, capital R, or lowercase r. In the pair, it's the same letter twice. Heterozygous, on the other hand, hetero means different or opposite, right? If you're heterosexual, you like the opposite gender or the different gender. So heterozygous is about alleles, and it means that there are different alleles for a single trait. And there's only one case of this, so that you don't need to worry about heterozygous dominant or recessive. It's just heterozygous. And that means in this case, you would have one capital R and one lowercase r. So this is a heterozygous. And these are both homozygous. This is just homozygous dominant, and this is homozygous recessive. So here, you're going to tell me, is capital H homozygous or heterozygous? Is big J homozygous or heterozygous? Is little p homozygous or heterozygous? Is big T homozygous or heterozygous? And then lastly, is big K homozygous or heterozygous? Answer below while you pause. Okay, the last thing we're going to go over is Punnett squares. So you saw in the video, they did Punnett squares with pictures, and it's super nice to do them with pictures, but we will pretty much just be doing them with letters as is shown in here. So the reason we do Punnett squares is because we want to predict the outcomes of when you cross two parents or when you breed two parents together. Because sometimes, like we showed with the peas, if you breed a yellow pea with a green pea, sometimes all of the offspring pea end up being yellow, and then you're like, what the heck happened? I wanted green peas, right? So the way that we do this is we're going to put the mom's genotype on the top and the dad's genotype on the side. And then we will be dragging the moms down twice and dragging the dads across twice and seeing the combinations that result. And you'll get 4 resulting combinations. And those are the possible genotypes of the children from this cross. So we're going to do an example, and I'm actually going to read through this one really quickly, and I'll show you it handwritten how I would do it. Okay, so this example here is hair color, right? Brown is going to be dominant over red. They're giving us that information, and they give us the mom as capital B, little b, and the dad is also capital B, little b, which means that they both have the dominant color of hair because they both have one dominant letter. So we're going to do a Punnett square after we answer all these questions, and then we will figure out what the possibilities of their kids are going to be. So let me change the camera. And we're going to change it to my dot cam. So the quality is a little iffy, but basically what this saying in the problem is that we know that brown is dominant over red for hair color. We know that the mom is capital B, little b, and we know that the dad is capital B, little b. So what color do the parents have? Well, if brown is dominant, that means brown is going to be capital B and red is going to be lowercase b. And if each parent has a capital B, that means that they both have brown hair. So mom has brown hair, and so does dad. And again, like I said, the reason you know that is because both of them have a capital B. And you only need one capital B to have brown hair because capital B is dominant. Are the parents heterozygous or homozygous? Now, remember, heterozygous means that they're different, and homozygous means it's the same. So homozygous would mean you have to have big B, or little b. And for heterozygous, there's only one possibility, which is big B, little b. Now look at the parents. They are big B, little b for the mom, big B, little b for the dad. So they are both heterozygous. So that's how you would answer that question. The last question would be, what must the genotype be to have red hair? Now, red hair is recessive, right? We know that red is recessive. So if you even have one B, you end up with brown hair, like the parents are. So if you want to get red hair, you have to have little b to show the recessive trait. Okay, so now we're going to set up a Punnett square. It's always useful to have scrap paper or space in your notebook ready so you can write these out. If you try to do these in your head, you're going to mess up. Even experienced scientists make mistakes, and it's too easy to mess these up, and it's too easy to do these right. So there's no reason for you to not draw them out and then get them wrong because you chose to be lazy, okay? So you know that the mom is capital B, little b, and the dad is capital B, little b. So we're going to do a cross between capital B, little b, and capital B, little b, or a heterozygous cross. It's called a cross because there's literally a cross or multiplication in the middle. So like I said, we're going to put the mom's alleles right here. So the mom is going to be capital B in one box and lowercase b in another box. And then over here, we're going to put the dad's. And the dad also has a capital B and a lowercase b. And the way this works is we're going to bring this b over here. And we're going to bring this b down into this box right here. And we're going to multiply them. We're going to bring this b down to this box, and we're going to bring this little b over, and so we're going to have capital B and little b. We're going to bring this b all the way across, and we're going to bring the little b down, and so we should have a capital B, little b in this box. And then lastly, we're going to bring this little b and this little b over, and we should have little b in this box. So those are the possible genotypes of the offspring, right? These are the results we're looking at the offspring now, or the babies. And so the genotypes, how many did we have of big B out of the 4? We only had 1. How many did we have of big B, little b? We had 1, 2, 2. How many did we have of little b? Well, we had 1 out of the 4. 1 divided by 4 is 25%. 2 divided by 4 is 50%. And 1 divided by 4 again is 25%. These should add up to 100%, right? Because this is the total 100% of the possibilities. Now we want to look at the phenotypes. So if you're looking at phenotypes and it has brown hair, that means it has at least one capital B. Which ones here have one capital B? Well, we see 1, 2, 3, but not this one. So we have 1, 2, 3, 3 out of 4, which is 75%. And which ones would have red hair? Well, that would only be little b, which is 1 out of 4, which is going to be 25%. So basically, the reason why we did this is because out of 2 parents that both have brown hair, each time their kid is born, there's a 25% chance that that kid will have red hair. But most likely, 75% of the time, it's possible that the kid born will have brown hair. So these parents shouldn't be surprised if they have a kid that has red hair, but they should know that it is a lower chance of having a kid with red hair than with brown hair. And so that's how you would solve this problem. So let's go back to the table now, and we're going to do another example now, and that's going to be about freckles. So when we talk about freckles, we're going to say freckles or no freckles is the trait, and we're going to be told that freckles is dominant. The mom is going to be capital F, lowercase f, and the dad is going to be lowercase f, lowercase f. And we're going to answer these questions. Which parent has freckles? How do you know? Is the dad heterozygous or homozygous? And then we'll do a Punnett square to figure out the possibilities of what their kids will look like again. So, like the question gave us initially, freckles or no freckles is the actual trait. And we know that freckles is the dominant trait, which means that freckles is going to be capital F, and no freckles over here is going to be lowercase f, right? Because the dominant is always capital and the recessive is always lowercase f. Now we're going to look. They gave us the mom's genotype, which is capital F, lowercase f, and they gave us the dad's genotype, which is lowercase f, lowercase f. Now the question first off asked us, which parent has freckles? Well, freckles are dominant, so we're looking for which parent has a capital F. And out of these two, only the mom has a capital F right here, right? So that means the mom has freckles. And how do we know? Her genotype is capital F, lowercase f, which has one capital F, which means it's dominant. Okay, now we're going to look, is the dad heterozygous or homozygous? Well, we're looking at the dad and he has little f, but then he has little f again, which means they're both the same, right? Homo, same, hetero, different. These are the same, so it's going to be homozygous. And we also know that it's homozygous recessive, right? So the dad actually in this case has no freckles, and the mom has freckles, but she's heterozygous for having freckles. Okay, so we're going to put the mom at the top now. The mom has a capital F, and she has a lowercase f. We'll put the dad on the side, and he has a lowercase f and a lowercase f. So now we're going to do the Punnett square together for this cross which was heterozygous, crossed with homozygous recessive. And so we're going to be able to drag the capital F down here with the lowercase f here. And so when we bring these two together, we're going to have capital F, lowercase f. We're going to take this capital F and drag it down here, and this lowercase f, drag it over here. And as we bring them together, we're going to have capital F, lowercase f. We bring these two together here, we're going to have lowercase f, and we bring these two together here, we're going to have lowercase again. So let's look at the genotypes of the results. How many capital Fs are there? Based on my count, there are 0, which is 0%. What about capital F, lowercase f? I see 1, I see 2. So this is 2 divided by 4, which is 50%. And how many lowercase homozygous recessives are there? We'll see 1 and 2. So that's again 2 out of 4, which is 50%. Okay, again, this total becomes 100% because it's 100% of the possible offspring. Now we want to look at the phenotypes. How many have freckles? How many have at least one capital F? That would be 1, 2, which would be 2 out of 4, again, which is 50%. If there were any of these, they would also be added in, but we had 0. Which ones have no freckles? So we're looking at the homozygous recessive, which would be little f. So we would have 2 out of 4, which would be 50%. And the reason why we always want to find the results of the genotypes and the phenotypes is because the questions on your STAR test and on any of your tests, honestly, in genetics is going to be asking you questions that talk about the result of the cross and what possible chances are their babies have specific phenotypes or genotypes. So back to the screen now. I'm going to ask that you do questions and answers on earlobes, which is going to be unattached or attached. As you can see in the picture, this one's unattached, so there's a separation close to the face, and attached means that there's no separation. It goes straight to the face and the side of the jawline. So unattached is dominant, and we're going to cross a homozygous dominant, capital U mom, with a homozygous recessive dad, little u, and he's going to have attached earlobes, and we're going to figure out the results, and I will ask you questions on them. So you should be prepared to do a Punnett square on your own for that. And then you'll do one with cleft chin, which is also known as butt chin, versus smooth chin. So no butt chin or butt chin, and actually having the smooth or no butt chin is dominant versus when you see a butt chin. So we're going to cross a homozygous recessive mom with a heterozygous dad. So you're going to do that Punnett square on your own on a scrap piece of paper, and then you'll answer questions about this and about the genotype and phenotype for me.